I will. Sorry. And actually, I will borrow the bell. It may help. Mogę? Okay. So I'm going to use it a minute before the end of the lecture, okay? There will be a small change in the schedule because Norbert asked us to be first one in this session because he needs to leave. So please join us and let's start the second session for today. But we need to change this. This is not yours, right? This is...
It's not.
Okay. Can you find your file? This one?
Yes.
Oh, sorry.
Use that.
Okay.
Thank you.
Can you join? Can all the speakers join us here?
Okay, so I will start. Thank you for the kind introduction and sorry for the change in the agenda. I do have to dash after this presentation because we are a dual-use company and we also operate on sports market. There is a huge conference by a Polish association in football and I have to present there also some dissemination of our work. So anyway, just a small remark, if you want to ask some questions after the presentation, of course I will be here and farther on you can contact me on LinkedIn. Okay, so I will present some more information regarding our experiment that was conducted on the current mission called AstroPerformance. So I would like to talk about leveraging AI for physical readiness in space missions. But first of all, I would like to start with a bigger picture and our motivation for leveraging this technology, this particular technology, and incorporating it into dual-use pipelines and also this space medical concept. So, as you are aware, we are now on exponential growth in terms of cargo missions, due to some lower prices to getting those cargo into orbit. And we can expect that with development of further technological issues and operational issues, the space tourism, the low orbit missions, the missions to the moon and Mars will also explode. So human missions will also exponentially grow in number. So that's a big opportunity, but also a huge challenge in terms of preparation and optimization of the current existing pipelines. And I think that AI could help us in those cases. So current preparations, I mean the whole mission preparations and taking into account like 6 months or 12 months working with astronauts and also even more with pilots and military critical services. That is an expensive, of course, process. And if we want to scale it out, we should think about incorporating modern technologies into those processes. So that is why our task was defined, taking into opportunity that we had this first Polish astronauts since a lot of years. So we could test the AI incorporation into the pipeline. So what we defined is that we wanted to use the AI engine that was trained on more than 3,000 athletes because we are also working with Sports Society. So we wanted to use this AI engine to perform a comprehensive assessment pre and post mission of astronauts that were involved in our project and also on the reference group. And then to use this AI to produce automatic reports on the state of astronauts and reference group and compare them challenge those results with experts to see if there's a value in those automatic reports, and to see whether we can optimize some amounts, some pipelines that are data-driven and data-dependent. So, in terms of the numbers in the experiment that we gathered, from all the measurements, we have received 14,800, more than 14,000 parameters that were grouped automatically by AI into more than 700 meta-parameters. And that was developed or assessed on actually 12 participants, but 24 studies. And we do have 4 experts from metabolical point of view, from motion point of view, biomechanical point of view, from structural point of view, so MRI assessments. So those experts are investigating the AI results. So we projected what we wanted to assess is to actually something that mimics standard studies for astronauts or also for military. So taking blood tests, MRI, we focused on the lower limb because that due to some constraints on MRI is pretty slow, so the whole scan of lower limbs take about 3 hours that we wanted to investigate. So the whole body scan in that kind of resolution would be too high number. That's why we focus on lower limbs. Also body composition and biomechanics plus cardio. So rather standard set of tests. And we wanted to perform that, and we performed that in pre-flight and post-flight assessment. So this is the whole mission preparation assessment, but we also wanted to focus maybe on some microgravital effects. That's why we involved the reference group. Of course, if you see the gap between the mission and 2-week mission, 2 weeks mission is pretty high, so we do not know that we can achieve the microgravity effect assessment, but we will search that in data with the comparison with the reference group. So we are at this stage that we could produce automatic reports and this grouping of parameters by the AI. So what we want to focus on is to, from this massive amount of data, get key performance limitators that could be a hint, a guide, what to focus on with the mission preparation ongoing. And actually, there are some pretty easy, low-hanging fruit that could be in somehow incorporated in the process, like drop of insulin on vitamin D or ferritin, that with some supplementation could affect better preparation of astronauts' health and could be taken care with ease. So that's just a preliminary results that are just in the data as a low-hanging fruits, but we are searching, of course, more detailed ones and more detailed relationships. In terms of motion analysis, we also managed to group those in that kind of radar plots, and we seen changes in stabilography, motor control, and force parameters, especially in jump performance. So that's what we want to focus on and investigate. In terms of structure parameters, so MRI and also body composition analytics, we did discover bone density related issues and MRI signal changes in the shin region. So that's what we want to also investigate further. So from the company perspective and what we want to deliver in this experiment is actually a proof of concept platform for integration of data that would incorporate also AI in the reasoning process. And that's what we want to achieve by the end of the year, maybe at the beginning of the next year. So how could it be used? For example, you can give a structure of critical services, military personnel that you want to monitor. You can easily integrate data that you are acquiring, some biomechanical data, some blood tests from data sources, some DEXA scan, or if you work with Google Sheets, that's also not a problem. We are actually now have 14 connectors to major data sources. You can also, because the processes varies between different preparation models, you need to use some sort of data cleaning tools, and it could also be automatized. So we use data lake concept to clear all the data to enhance AI possibilities to reason on those data. So that is also our concept that we are utilizing. And so automatized cleared data can be used and incorporated with AI with modern methods like chat, but this is our own LLM models that is trained particularly on musculoskeletal system. So to organize those data into modules that could be used. If you are working on neuromuscular data, you can ask for that kind of organization. If you are working on DEXA scans or other type of data, you can also use that kind of organization. So of course that was a simple example, but here is the structure that we use for this particular experiment. So 3 modules that aggregates data into meta parameters. Every meta parameters incorporates a set of number of detailed parameters with norms from more than 3,000 athletes that could be compared. And it is very important to incorporate AI into the process of particular space to work to limit model hallucinations. So that is why we are also open to collaboration to create a space environment, aerospace environment that could be also used to work with this model. Interpretation, also another tool that you can use when you have a nice organized data. And for example, if we have a group of or cohorts of specific critical service personnel that differs in terms of some physical factors. So we want to also explore what are relationships between those, between different parameters. So this could be done also by interacting with AI, not knowing any Python code or different type of languages that was previously used for exploring data in data science. So going to recommendation. And the third thing that I want to show is also ready, is recommendation for cohorts or for some specific sets of data. So for example, if you want to optimize for 5, 10, 15 groups of different type of preparation models, then you can easily explore the data and ask to use PCA or other components that is well known in data analytics, but you can use a chat component to do so if you have it incorporated in your processes. Finally, monitoring and monetizing. So the thing to optimize the data utilization. So you have some hypothesis in your data and you want to monitor it in time. So you would also need a tool to do so. Those are components that we are focusing at. And as I said, the deliverable of this project will be a proof of concept platform for integrating data in sports, but hopefully also in aerospace segment. So we foresee that the data integration could support doctors, engineers and trainers working with astronauts and also military personnel, and especially for personalization of mission plans with respect to health. Another thing is that mission is an accelerating aging model, so we can move also the results from the military segment to the insights for sports, longevity, rehabilitation. So this is the dual use that I was talking about. So, thank you. Right on time.
Thank you for the presentation. And now I would like to invite Alicja Trębińska-Stryjewska.
Good morning, ladies and gentlemen. It's my pleasure to present experiment Immune Multiomics, one of the 13 experiments performed during the Ignis mission. The full name of the experiment is Transcriptomic and Epigenomic Analysis in Peripheral Blood Mononuclear Cells following residence in low Earth orbit. Firstly, let's take a look at all the stressors that can specifically influence the immune system. And here we have microgravity that changes the mechanical unloading of cells, which might affect the cytoskeleton. And this is very important part of the immune cells, which have to have the ability to migrate and perform phagocytosis. We have radiation that can affect the DNA of the cells and also induce some oxidative stress. And we have different factors like confinement, closed environment that can enhance the level of physical, emotional, environmental stress, and it can in turn activate the hypothalamic, pituitary, adrenal axis and sympathetic nervous system, and also affects the immune system. And last but not least, we have the we can have the host, altered host microbiome interaction and the affected regulation of immune processes. So there is a body of work concerning the effects of this space flight on the immune system, both the innate and adaptive part. They were performed different models, the real spaceflight or the analog missions, and also humans, animals, and in vitro cells cultured in vitro. And there are several findings, some of them contradictory, actually. But we can say that you can observe chronic low-grade inflammation in astronauts. It's detected by the changes in metabolites and also the cytokine profiling. There is an increase in the number of circulating neutrophils and the neutrophil to lymphocytes ratio. But actually the processes that are important for phagocytic cells like phagocytosis and the oxidative burst in monocytes and neutrophils can be affected. And actually it might depend on the length of the mission. We also have some changes in the polarization of macrophages and conflicting results on the number and the activity of natural killer and dendritic cells. Here it's either the decrease of the number of cells and decrease of the activity, or actually there are no changes observed. We also have some changes in the adaptive immune system. And here it mainly affected the T cells. B cells are rather resistant, seem to be rather resistant to the space flight conditions. With the T cells, we have impaired development of them, reduced response to mitogenic stimulation, and this might be due to the lower levels of interleukin-2, which is very, and reduced expression of the interleukin-2 receptor on T cells, which is very important for the activation. The decreased performance of the cytotoxic T cells is observed in the form of reactivation of latent viral infections like herpes viruses. It's actually usually at the subclinical level, but we can detect some shedding of the viruses in the urine or saliva of the astronauts. Okay, so we wanted to look at the molecular changes in immune cells because the changes at the gene level, at the gene expression level, can tell us what exactly is happening in the immune cells. And in our experiment, we chose to concentrate on gene expression at the mRNA level. So this tells us which genes are active, which are not, and what processes are activated or repressed in the immune cells. But we also want to look at the DNA methylation. Here it was already mentioned, DNA methylation. It's the addition of the methyl group to the DNA strand, and it affects the chromatin status when the methylation is located in the promoters of the genes. It induces chromatin condensation and decreased binding of the transcriptional factors, so the transcription is turned off. And observing such de novo methylation events can tell us about the potential long-term changes in the gene expression. When we started, when we applied for this experiment, there was actually only a few studies on gene expression in peripheral blood, mononuclear blood cells, which are basically lymphocytes and monocytes. One seminal paper, NASA twin study, which looked at the two subjects, but they were monozygotic twins. Flight subject that stayed at the ISS for a year, and his twin brother that stayed on Earth. But that was a long-term mission. But there were some changes detected in the PBMCs related to the immune functions, and not all of these gene expression changes return to normal after 6 months after mission, and also the general level of DNA methylation was not that changed that much, but some of the changes related to the promoters of the genes that regulate T cell differentiation and activation. So that was quite interesting. And the second work was done on a 6 months mission, and here also with a bigger group of the astronauts. It also detected some changes in gene expression in immune cells. So our scientific questions were that whether we will observe gene expression changes during a short mission in PBMCs, or maybe after the return to Earth, and whether these changes persist after the recovery period, and are there any changes in DNA methylation status during this short-term mission, and can we correlate this gene expression and DNA methylation changes? So to do that, we designed this study when we plan to collect blood samples pre-flight 1 month before the mission. We wanted to have a later time point just before the mission, but we didn't get the green light on that from ESA because collecting blood during the quarantine was technically difficult. 2 time points to blood collections at the ISS, and then 3 blood collections afterwards. The last one after one year after the return to Earth. Here we wanted to concentrate on the mononuclear cells, isolate DNA, RNA, and then perform gene expression DNA methylation. To do that, we used specially designed blood collection tubes that allow for separation after the centrifugation, allow the separation of PBMCs from the red blood cells. The samples were treated the same, the ones that were collected on Earth and also at the ISS. We used the abilities of the human research facility in the Columbus model that includes the centrifuge. You can spin down your samples, and the samples were frozen. This might not be the optimal protocol, but it was actually used before in the 2 studies that I presented to you briefly. It allows us to store the samples for a long time, and actually that's quite useful. I will tell you in a minute why. And we need to make sure to preserve the cold chain during all the transport of samples from the ISS and on ground. So here is my favorite picture, and actually that's the only one that we had so far from the International Space Station, because it shows our tubes prepared for the blood collection. So far, there were 4 blood collections pre-flight. Due to the delay of the mission, it was not exactly as planned, so it's 2.5 months before the mission, not a month or half a month, but we could not do anything with it. Then we have 2 in-flight blood collections and also the first post-flight blood collection within the first week after the return. It was all well designed, and all the temperature and time constraints were met. The ones that we planned in our activity requirement document. So the in-flight blood collections were done with flying colors. Unfortunately, I'm not able to present any results from this experiment because we are still waiting for the samples to return from the ISS. And the wait is longer than we expected, because the last news was that they might be back in March 2026. Obviously, we are a little bit worried about the condition that they were in, because because that might actually affect the cells. And we need to collect the samples 6 and 12 months post-flight. Okay. And perform all the analysis to avoid batch effect. We want to do it all in the same time. And actually, it's it's becoming a really hot topic the last 2 years. That works new publications regarding this topic of changes of DNA and epigenomic markers in mononuclear blood cells. Last slide, I would like to acknowledge my team. Here a picture taken with Sławomir during the briefing of the mission. As you can see, there is not many of us, so I really don't know how we've managed to do it all. And thank you for your attention.
Thank you. And now I would like to invite Elżbieta.
Good morning, everyone. It's my pleasure to present you the flow of our experiment, which operational name was Human Good Microbiota. But below you can see the full title of our experiment, which indicates that the main technique which we used were sequencing. It's a quite modern technology of, let's say, recognition of the bacterial species which can be detected, cultivable and uncultivable at the same time. We did it in Biomedical Engineering Center at the Military University of Technology. And short timeline, kickoff meeting was held on April 2024. Launch, the date of launch and the undocked of the Axiom 4 mission you certainly know. It was in June and July this year. And our last BDC samples, BDC, it means baseline data collection samples, arrived in November 4. And as you can see from those days, we were unable to do all analysis yet. And I will present only preliminary data here. Just a short technical overview to present you some, let's say, difficulties we met. The download vehicle was Axiom 4. Our samples arrived on Earth, and I am very happy of it. And it had to arrive at very deep freezing conditions. So we use onboard and the download freezers. The number of subjects involved in our experiments was 2 astronauts, which spent over almost 5 hours of their time on ISS executing our experiments. And of course, the main topics here were medical board approvals. Thank you very much for your Institute Medical Board for helping us in getting this medical board approval, which was the most important step in the execution of this experiment, because further was ESA and NASA, but without your help, we won't be able to do anything. And of course, baseline data collection was required. It's our team. It's more people than in my colleague team, but still not so many. And I would like to mention here our very important collaborators from ESA, Jutta, Tiziana, Laura, Alexandra from French, which help us in technical issue. Very friendly and nice team from Cologne, sorry, it's in Polish, Maria, Petra, and Edwin, and also Michelle Griffin from Axiom Space in Houston. They help us in collecting the BDC samples. And of course, the most important person, Slavoj, who governed execution of our experiment on ISS and was mainly responsible for a very, let's say, smooth flow of this experiment. What was the idea behind? The microbiota, what's the microbiota is? I am certain everybody knows some data about this phenomenon, but just to mention, it's a very, very numerous population of bacteria, fungi, viruses, which inhabit certain and specific parts of the human body. We specifically was interested in gastrointestinal tract. When something wrong is going with our health, the state of dysbiosis occurs, which means we get some microbial imbalance within the microbiota. Microbiome is very similar terms, which means it might be a reduction in the number of bacterial taxa or very significant shift in proportion of good bacteria versus bad bacteria. I will show you this in a moment. Looking at this problem from the very scientific point of view, you can see that this good microbiota is responsible for the intestinal barrier function, for digestion, immune system function, proper metabolism, protection against infections, and also drug metabolism. Many factors can influence the well-being state of microbiota, and among those factors, the most important for us in our experiment was stress and changed dietary habit, which occurs during the mission. And the very important point also is the connection between good microbiota and let's say, good activity of the brain. Okay, when this connection is broken, you can observe some neurodegenerative disease or some psychiatric disorders. It is important in terms of space mission. To study this, we had some very detailed objectives, which can see you here. We wanted to observe some, let's say, BDC samples 3 months before start, 7 days before start. We wanted to take 3 in-flight samples and also to monitor the microbiota after return to the Earth, post-flight, 7 days post-flight, and 90 days post-flight. Looking at this timeline, you can see 2 very important points. One, 7 days before flight, and the second one, 7 days post-flight. This point is the first time which was performed in this schedule, because in the previous experiments performed on microbiota in the space, this point was never monitored because NASA refused to get samples during quarantine, as my colleague mentioned. This time we had a great discussion, and finally I was allowed to get those samples from quarantines. They are very important because they are very close to the start and may present some stress-related changes which can occur during this experiment. What we expected? We expected some shifts in microbiota composition. For example, we were interested in some pro-inflammatory bacterial taxa, which are the first sign of dysbiosis. And of course, long-term project objectives were how to maintain a healthy microbiota under conditions of extreme isolation during spaceflight. We this project has several constraints, as you can see. The most important were the requirement to keep all our samples in very low temperature just to preserve the bacterial composition. The bacteria multiply every 20 minutes. It means that our samples had to be frozen immediately. It's easy to say, but it's not easy to execute being on ISS, okay? When the time between the facility when the samples are collected and the facility when the freezer is not so close, okay? They have to run through the ISS within 20 minutes, and I was told that it seemed like a, let's say, athletic exercise to execute this time efficiently. The other constraints was the unload of the samples in the same temperature and let's say transportation in a very low temperature to our facility in Warsaw. It was done perfectly. I am very happy of it. We took so many samples, and to my astonishment, we took 2 times samples during quarantine, which is, as I said, the main, let's say, important point in our project. 2 astronauts, different times, of course, because they are independent. The last, our samples were taken in November. All of those samples arrive in our laboratory in very good conditions, in very proper temperature, and we are very happy of this because they are, let's say, valid from the scientific point of view. Now we start to analyze those samples. The analysis is by sequencing. The sequencing is done by NGS technique. It means we observe synthesis of a new DNA strand by just fluorescence signals, which are recorded by the sequencer and then translated into the DNA sequence. The most difficult part of this analysis is bioinformatic analysis, which has to be done by ourselves. It has several steps like a quality control, taxonomic assignment, abundance calculation. Abundance is the proportion of some specific taxa in all bacterial populations. I am very happy because we have already those pipelines of bioinformatic analysis ready to use, and just we are waiting for some medical board agreement, which I mentioned in a moment. These are our preliminary results, and as you can see here, they are all top 8 taxa in each astronaut. It's a before flight, the first BDC samples, and those 2 astronauts has completely different proportions of those 8 dominant taxa. The first astronaut, please look here, has over 16% of Bacteroides. It's a bacterial genus which is pro-inflammatory. It's a very bad sign of a microbiota health. The second one has very low level of Bacteroides, but very high level of very good, let's say, like probiotic strains. Probiotic, it means healthy microbiome. So I was very happy, we were very happy that those, let's say, randomly chosen astronauts has completely different start point, and then we can follow the let's say, changes in this strain composition. I know what happened later, but I cannot present because I don't have medical board agreement yet. But when this situation will be confirmed through all those samples we took, what it can mean. It can mean that probably the composition of diet for each astronaut should be individualized to preserve the health of this astronaut. We have to push for the persistence of the proper and healthy taxa in the microbiomes, and it can be done probably by personalized probiotic supplementation. Okay, still pending. Those are data we are still waiting for. Diet logs for ISS, medication logs, and supplement logs. And you can see there is a terrible word here. It's desired. It's not required. The word terrible desired was, let's say, executed by ESA in our project. It means I might not have those data because they are not required. All our colleagues sitting with me and smiling now knows what it means. Smiling, not smiling, sorry, crying. And I look here at our colleague from Polsa, maybe you can push on ESA to provide those data, because all physicians here, all medical staff here knows what those data are for. And of course, we still wait for approval from medical boards, and that is the reason I cannot show you results, because we collected more samples than we were supposed to during the flight delay, okay? And also during the kindness of the second astronaut who provide us extra samples. And I have to proceed with ESA Medical Board, with NASA Medical Board, once again, to get the approval to use those samples in my analysis. Okay, who can benefit? Please read very shortly. Very, very different groups. And thank you very much for your attention.
Thank you. Now I would like to invite Jakub.
Thank you. Good afternoon. Ladies and gentlemen, I'm very happy that today I have an opportunity to tell you about the experiment we are carrying out on the board of the International Space Station. The experiment is called Stability of Drugs. At the beginning, let me introduce our research team. We are scientists from the Center of Polymer and Carbon Materials of Polish Academy of Sciences from Zabrze. Also, we are members of the Laboratory of Polymer Biomedical Materials, and in our everyday work we are focusing on the new applications of polymers in the field of medicine and pharmacy, and among the others we are developing bioresorbable polymer drug delivery systems, which are main research objects in the Stability of Drugs experiment. I think you will agree with me that the health of astronauts is one of the most important, maybe the most important factor ensuring success of the mission in space. During the space flight, especially a long-term space flight, bodies of astronauts are under the influence of several factors, like increased level of cosmic radiation, low gravity, feeling of isolation and stress situations, and also closed environment. And these factors may cause some negative symptoms or diseases leading to deterioration in health. So here we have following situation where continuous effects of negative factors may lead to recurring health problems affecting both physical and mental health. And we propose a solution in the form of constant supplementation or pharmacotherapy
to reduce these negative effects or to prevent them. However, storing drugs in space is quite problematic because, as we know from another research, drugs stored on the low Earth orbit exhibited accelerated decomposition in comparison to the drugs stored on the surface of the Earth. So we need to protect the drugs. And fortunately, some materials, especially those made of light atoms, like, for example, carbon, oxygen, hydrogen, have the ability to partially block the cosmic rays. So we could use them to protect drugs. Unfortunately, our polymers we are using in the experiment are made of these atoms. So our idea is to use polymers in two functions. The first function is to protect drugs from the cosmic rays, not by forming some kind of containers or plastic pills, but by mixing polymer and drug at the molecular level. We did it by just dissolving these two components and form such simple foils. Foil is good in the basic research because we have eliminated here the geometrical variable to our results. The second function of the polymer is to control drug release. We can control drug release by two factors. Choosing polymer of desired properties, like, for example, hydrophilicity, and to process it to specific derivative. For example, it could be nanofibers, microspheres, nanoparticles, whole implants, coatings. We have chosen simple foils. Thanks to these two factors, we can control the time of the release of drugs from days to even weeks, and also the release doses. So we have control over the drug release profile and how such system is working in the biological environment. After introduction to the body, water from the body fluid is penetrating the polymer carrier, dissolve the drug, so the drug molecules can be released to the surrounding tissues or blood, and simultaneously water is reacting with the polymer, and due to the reaction of hydrolysis, they are forming specific low molecular weight byproducts which can be metabolized by cells, or when it's not possible, then they can be easily removed from the body. So we have here full absorption of the polymer, and this material is not accumulating in the body. Let's move to the experiment. Stability of drugs is very simple shelf life experiment. And to cut the long story short, we have prepared our samples in the form of drugs, polymers, and polymer drug delivery systems. We have formed our payload that was sample packages, which has been sent to the ISS. And they are going to be stored there under the influence of increased radiation, cosmic radiation, for 3 years. So our results, full results of the experiment will be available, I think, at the end of the 2028. However, every year we are going to download some of the packages and test them in our laboratory in terms of changes in chemical structure and physical properties of both polymers and drugs. We also want to check in vitro cytotoxicity of polymers exposed to the cosmic rays for such a long time. And also we want to compare drug release profiles for samples stored on the low Earth orbit and in our laboratory in Zabrze, because we are carrying out a simultaneous reference experiment in our laboratory in the same temperature conditions. And we have two conditions. Ambient temperature, it's about 23 Celsius degrees. These samples are stored in the Columbus module, and lower temperature in the cold storage facility in +2 Celsius degrees in Kibo module. So here we have two conditions more aggressive in ambient temperature and more mild where lower temperature helps to slow down chemical processes. And also case of the cold storage is reducing further the cosmic rays. So this is maybe it will be enough to put our samples to the fridge and we will get better results. Uh-oh. Houston, we've got a problem. Oh. Thank you. As regards our payload, a single package with samples contains two types of reference samples. It's pure polymer and pure drug. We have three types of polymers differing in the rate of biodegradation and physical properties, like, for example, crystallinity. And also we have 6 types of drugs. Drugs are differing both in chemical structure and biological action. And finally, we have 18 types of polymer drug delivery systems, which are the main research objects. Each sample has been secured for the flight and for the storage by multi-layered packaging made of heat-sealable polymer foils packed under vacuum. To sum up, I would say that our experiment stability of drugs, it's a kind of pioneer research because, as far as I know, we are first to test the polymers, medical grade polymers as a drug carriers for the application in space. Also, the nature of the experiment is a basic research, and this is a very comfortable situation because every result, whether it's desired or not, have significant value, scientific value for us. We are going also to answer some questions from which the first 2 are of the key importance. We want to assess the potential for using polymers as drug carriers for the pharmacotherapy during this long-term space missions. And also, we want to answer the question whether it's possible to extend the stability of drugs by mixing the bioactive substance with polymer at the molecular level. And at the end, I'd like to highlight that polymer drug delivery systems can be beneficial for each of us, especially in therapies when we need to administer drugs for prolonged time, and also in such situations when we have low number of medics and large number of patients, like, for example, at war zones or areas affected by natural disasters. So it's worth to develop the science in the field of polymer drug delivery system. And at the end, this is the explanation of our patch. Thank you.
Thank you. And now I would like to invite Eva and Franco. Please join us here on the stage.
Thank you. Welcome, everybody. It is very nice to be here. And I mean, we are so happy that we can present our experiment, and not only because we are here, and that's why we are so glad that, I mean, we got invitation for this conference, that we would like to let you know who we are, and we would like to share our interest with you, and we are open for the collaborations because we would like to be immersed in the community with the same interests. So, I mean, I'm Ewa Szuszkiewicz, I'm professor at the University of Szczecin, and I'm astrophysicist and astrobiologist. And I just mentioned a few things which are relevant for this conference here. So I am a chair of the division of the astrobiology and space medicine in a committee on space and satellite research of Polish Academy of Science. So please do, take activity and join the initiative of our committee. It's very important that we work together. And I am astrophysicist, so of course, I am involved in the mission which are looking far further than our solar system or our planet. So I am involved in the mission Plato, which will discover more planets around other stars, and also Ariel, who will say if this atmospheres around new discovered planet are they similar, different, or if there could be a life on this planet. So also X-rays mission is not far from my interest. So New Athena is also there. But now let me give the floor to Franco.
Okay, this is not necessary. I'm Franco Ferrari of the University of Szczecin, and I'm a physicist. Usually I'm working on polymer physics, so the previous experiment on polymers has all my sympathy, even though I'm working in some bit different subject. I'm also in astrobiology. My interest in space and space exploration came from my participation as a member of the Cosmic Study Group of the International Academy of Astronautics, where we studied we gave recommendations for the career dose for the astronauts, in particular to limit this dose to a safety level. So, okay, so what we do or what we did. So first, ionizing radiation. We had an hypothesis that ionizing radiation can shape the evolution of life on Earth. And this is, for instance, that there are three main arguments about that, which you can find in the paper listed there. One is that there is a deep biosphere in the Earth between 10 kilometers to 21 kilometers deep in the crust of the Earth. And some of the bacteria that are living below are the same that the bacteria that live on the surface, and their response to ionizing radiation is the same. And there are other two evidence that are, of course, sometimes so maybe coincidence, that show that actually ionizing radiation from the cosmic from cosmic rays could have shaped the evolution of life on the Earth. And then we gave a recommendation to mitigate against human health risks incurring due to, for instance, solar energetic events, like, for instance, solar flares. We see here some example, and these are unpredictable. So the recommendation was to give to astronauts in missions information about the space weather, so that they could be prepared, reorient, for instance, the spaceship in such a way that the damage will be minimal, or to create a shelter, to build shelters inside the spacecrafts in such a way that if there is some emergence, the astronauts can go fast and get a shelter. So this is.
So now, I mean, it's TardigradeGene. That's our door into the community we are in now, and I'm PI of this experiment, but this is done in the collaboration of 3 universities: University of Szczecin as a coordinator, Adam Mickiewicz University in Poznań, and University in Silesia, of Silesia in Katowice. Our aim in our experiment is to answer the question, can tardigrades help in protecting other organisms in space? So in brief words, what we've done, we modified yeast using the gene of the tardigrade in order to make the yeast stronger. So then all applications which are already very wide can be even better, and I will just come into this in a while. However, that's what I would like to mention here is why tardigrades. Tardigrades are the extremely strong organisms. So if they can survive and deal with this different, very very bad condition which occurs in space, so why not yeast? Of course, yeast was chosen also because it's an organism which is relatively strong. So, it's not really very, very weak, but, it's still not enough for the space conditions. So, I will not go into details of the experiment. We covered this very widely last week in Warsaw, but I would like to make a point about the prospect, what will be the outcome of this and where to go further. Because, experiment is still going on because we've got already the material from the International Space Station. However, without the ground control experiment, we cannot get the full conclusions from our studies. So the ground-based experiment is still going on. So please stay tuned. We'll have results probably in the middle of the next year. And what are all this for? So, genetic engineering methods which are used in our experiment prospect for the building and maintaining the ecosystems on our future missions to the moon or to Mars. And also for the long travels and for this ecosystem, for us being on these different bodies, not on our planet, we need the energy, we need food. So yeast is a wonderful organism for that. We can produce biofuels, and yeast for a moment is the best organism which is studied for the space food system, so that you don't need to bring the food from Earth, but you can produce it on site. And very important for this conference as well, application, and the yeast is a very good practical model to study effects of space condition on human health. That's what we are all for here. So, for some particular questions, you don't need to do experiments on humans, but you can do these experiments on yeast, especially as far as the ionizing radiation is concerned. And we have a dream. So what we dream of? that's sequence which you are familiar with, a strategy from ESA. And in our perspective, so we would like to go, our dream is to go to Mars. But there are many obstacles, and you all covered this very nicely during your talks here and the questions as well. But I would like to focus on two of them. One which we studied on ISS, so microgravity. So you see the numbers over there that you have 1g, g is the Earth acceleration of gravity, and on ISS is almost zero, let's say practically zero. On Moon is 0.17 g, and on Mars is 0.38. So I mean, there are different conditions. So you have microgravity and you have reduced gravity, which is also very important. But the most interesting for us, at least, as you already heard from Franco, we are working on ionizing radiation. So it's at the moment the most difficult and obstacle to go to Mars. So on Earth, we have 2.4 microsievert per day. On ISS, 720. On the Moon, 1,368. Mars, 696. So, of course, there are measurements from particular periods. So, I mean, they are not always like that, right? But you see the problem here between the Earth and what we are going to find on our places we are going for. And now I give floor to Franco again.
So I'm lucky here that the talk of our chairman already has introduced all this stuff. So the detecting of damages made by radiation. And in fact, our dream is to detect the onset of cancer, which is not a trivial task in the human body, using the method of precision oncology and molecular medicine. So we believe that this is not the only way, of course, but it is a good point of view that helps very much, because what predicts precision oncology, this is the idea that there are driving events, so mutations at the level of DNA, and on the other side there are other events that can accelerate that mutations, the number of mutations, and these are all these other effects, like for instance, it's called epigenetic effect. And organoids we propose as a possible model. So we have to finish organoids, one should say, that have some advantages, but also several disadvantages. I have no time to speak about that. And we have an ongoing collaboration with the Wrocław Medical University to make organoids on chip. And the collaboration plans with the Pomeranian Medical University of Szczecin. And of course, we are open to other collaborations. So thank you very much for your attention.
Thank you for sharing us with your results. Now we have time for a short discussion, so please, maybe you can join us here. Are there any questions? Speakers. Okay, so I would like to ask you all, actually all of you, how hard it was to organize the payload for the.
Maybe I start. It was very hard indeed, especially that in our case, we prepared two different versions of the payload, because there was no guarantee that there will be space sufficient in the cold storage for our biological material. So I also would like to thank here POLSA and ESA and our ministry that, I mean, they warned us that, I mean, to be really on the safe side, you better prepare to a smaller version, so the one which gives you valuable scientific results, but do not go for really max, because then you can have a situation that you will you will have nothing, so it will be no place to fly. So I mean, that was the main difficulties in our case.
Preparing our payload was difficult, but I think not as difficult are, for example, your payload, because our experiment is very simple, and we have chosen also these drugs that already have been to the ISS, so the procedure was simplified for us, and I think it went very smooth.
Thank you. I prepared, of course, my payload, but I was very happy and lucky one. After a long discussion with NASA, they agreed to, let's say, borrow or allowed to use their kits for data, for sample collection. Therefore, I let's say, avoid some kind of security check, because my kits were already on ISS from the previous experiment, and I was allowed to use it. It was a good idea. Otherwise, the sample collection, I mean the stool collection, wasn't so easy, because it's a very hard, let's say, mode of this collection due to the specific technical constraints, okay? Not only regarding collection on the human side, but also from the technical side, okay? But those NASA decision was very helpful, and therefore, especially for astronauts, because it was much easier for them to collect those samples. Yeah.
In our case, the magical word opening the doors was heritage. So that's why we decided to use the blood collection tubes that were already used on the ISS. So we knew there was all the safety baseline documents, and so I would say it was not that difficult. One thing that we need to observe from the safety point of view was the release of glass fragments, because those tubes are made of glass. So we had to wrap them in a tape. So that in case the break in microgravity, the glass fragments are not released and present hazard to the astronauts or the equipment. We also took advantage of some of the equipment stored at the ISS, so we only had to upload the blood collection tubes, but all the needles and I don't know, the plasters and stuff like this were taken from the human research facility. So that was also very helpful for us.
Thank you. If there are no more questions, no, it was a question. So if there are no more questions, I just want to let you know that there is one another slight change in the schedule, because right now I would like to invite you for dinner, not after the next. So I hope it's a good change. And with that, I want to thank all the speakers. Thank you. Thank you.